Okay, we're 5 minutes early. Is that okay for everyone if I just start? Yeah, I thought so. Now, we have 2.5 hours. I am sure you're all going to love, right? So, I will do the first lecture in about, I don't know, an hour and 20 minutes, might be a bit less, might be a little bit more, where there's a good point to break off. And we'll all have a break for 10 minutes and come and do the second half later on. So I think the plan is going to be, the basic trophic types, a little bit about, physical chemistry of metabolic energy, and then we're going to go into the different ways bacteria can generate metabolic energy, starting with fermentation pathways, then respiration, and then weird stuff. Okay, and yeah, so that will be enough for anybody. Okay, so for the trophic types, you might all have heard about these terms. So bacteria are categorized by what they use as an energy source. If they use photosynthesis, then they are phototrophs. If they use some kind of chemical starter as the source of their metabolic energy, they are chemotrophs. Related to this, but not always the same thing, is what is the source of electrons. Either it's organic carbons, in which case you call the bacteria organotrophs. If there's an inorganic source of electrons, then they are lithotrophs. And then finally, carbon source. If they use organic carbon, then they are heterotrophs. Whereas if they use inorganic sources of carbon, bacteria are designated as autotrophs. So you have 3 different binary categories here. So there should be like 8 categories, 8 different trophic types that are theoretically possible. But in fact, there's only really 4 that exist. Okay. So here we start off with, phototroph, chemotroph. Each of these categories is divided into 2, organotroph, lithotrophs. And then theoretically, you might be able to divide all of the phototrophs into 4 categories and all of the chemotrophs into 4 categories. But in reality, pretty much these are the only ones you have. So if you have phototrophic bacteria, everybody got these slides, by the way? Yeah. So if you are a photoorganotroph, that means the bacterium generates energy by photosynthesis, but it uses organic carbon as a source of electrons. So organic carbon is available to these bacteria. And of course, they will use it also as a source for their carbon metabolism. So the photoorganoautotroph category does not exist. Okay? Similarly here, if you're a photolithotroph, that means you don't have any organic carbon to use as a source of electrons. So there would be no point in being a photolitho heterotroph, because if organic carbon is available, then they would use it, these bacteria would use it both as a source of carbon and as a source of electrons. Okay, so that's why these categories are empty in both situations here. So really there's only 4 trophic types for bacteria. Okay, photolithoautotrophs, photoorganoheterotrophs, chemotroph, litho-autotrophs and chemo-organoheterotrophs. So there's some redundancy in these categories. And to give you a couple of examples, okay, so autotrophs, cyanobacteria, they're phototrophs, they have light as a source of energy. The electron source is water, and the carbon source is CO2. Nitrosomonas, it's a chemo-autolithotroph. So there is an inorganic source of electrons, which is also the source of the energy metabolism, and they are carbon fixing. Okay, so chemical source of energy, inorganic source of electrons, and inorganic source of carbon. And then there are most of the bacteria that we will grow in the lab, heterotrophs. We need to give them sugars, peptones, some kind of organic carbon, which will be energy source, carbon source, and electron source. Okay, just to make sure everybody has got this correctly. Okay, the other thing is, don't mix up this autotroph, heterotroph stuff with what we saw the other day, auxotroph, prototroph. Okay, so let's just see if you get this correctly. Has everybody made up their minds? Okay, so who says this bacterium is a photolithoheterotroph, prototroph of biotin? Nobody. Second answer. Who said that? Oh. Third answer. Who says this one is right? And the fourth answer? Nobody. Hang on. So we have about 4 people who have decided, and everyone else did not give an opinion. So would you like some more time, everyone else? Yeah? Okay. So think about what these terms mean. What does prototroph, auxotroph mean? And if you're hesitating, tell me which 2 you are hesitating between. Okay, this time, are you all ready? Okay, I'll give you this. Are you really ready? Not so sure, right? Okay, answer A. Anybody want A? Answer B. Anybody want B? Answer C. Anyone want C? Yeah, most people. Answer D. Nobody. Yeah, is that right? Yes, that's correct. Okay, photolithotroph, that's easy. Auxotroph means it needs biotin, cannot synthesize it. You all got it right. Good. You'll be amazed how many people do not understand this. Okay, so now before we look at the metabolic pathways, I want to take a little bit of time to go over what do we mean by metabolic energy. And you are probably seeing this kind of stuff at the same time in the enzymology course, right, with Mr. Hoffman. Oh, which starts tomorrow. Okay, right? And so this will help you get ahead of the game in that case. So we'll go over this one by one just to make sure uh, everybody understands why some metabolic pathways are possible and others are not going to work. Okay? And we have to step back a little bit and think about thermodynamics and physical chemistry. Okay, so it all depends on the Gibbs free energy of a reaction, which has two components, right? So delta H, what is this one? Enthalpy, right? So this is the heat term, whether your reaction will release heat or whether it requires heat input, okay? And this one is entropy, yes. Very useful. This one is temperature dependent. This one is not. So the entropy term will be more important as temperature increases. Okay, so exergonic reactions will have a favorable delta G. So delta G will be negative. Endergonic reactions, delta G is positive. That means you need to put energy in to make the reaction go in that direction. Okay. Now, of course, in cellular biochemistry, a lot of the reactions that a cell will need to perform are endergonic. They require an energy input in order to, for example, protein synthesis, synthesis of polysaccharides. You need to put energy in. So how do you do this? Well, you have to kind of couple an endergonic reaction to something which is exergonic. So overall, the whole process will have a negative delta G. Then it will work. Then your reaction will go in the direction you want. And often, the energy source is ATP. And you'll have hydrolysis of ATP to adenosine triphosphate to adenosine diphosphate, liberating inorganic phosphate. So this works because the hydrolysis of ATP is exergonic, right? Now, one of the things that people don't understand, or it's not clear often, is why this is the case. Why does hydrolysis of ATP liberate energy? Okay, so I'm going to give you once again these 4 choices here. Think about which one of these seems to be the correct explanation for you. Well, for anybody. yeah, that's the way I learned it, actually. Okay, everybody made a choice on this one? Okay, so who says 1? It's a high energy bond. What's the hands going up? Who says 2? Because negative charges are separated. No. Because phosphate groups are good electron donors? No. And because the biochemistry textbook says so. A couple of 2 or 3 people being honest here. So everybody points out this one. Yeah. And I understand why, because that might be the way it's taught. But I don't like that explanation because it's a circular argument. What is a high energy bond? It's a bond that releases energy when you break it. But why does it release energy when you break it? It's because it's a high energy bond. You see, this is a circular argument. It's not an explanation. It's a reformulation in different words of the thing you're trying to understand. Okay? So this, to my mind, is not the correct answer. or at least it's not an answer that tells you anything useful, all right? So actually, I remember this is what my biochemistry tutor explained to me in a tutorial way back. Look, it's very simple. Here you have a lot of negative charge on atoms that are very close together with this triphosphate group here. Everybody knows electric charges, they repel each other, right? So when you have a lot of negative electric charges in very close proximity, it's unstable. And if you can separate these electric charges, it's going to be very energetically favorable. So for me, this was a real explanation why breaking this bond liberates energy. Okay. So that's why I think this works. Now, what does a cell need ATP for? Well, this is from a microbiology textbook. All these different processes that a bacteria are going to use, their objective is to generate ATP because that will allow the bacterium to perform chemical work, synthesize molecules that are interesting. Transport work, ABC transporters, importing molecules against a concentration gradient. They require hydrolysis of ATP for this process to work. Mechanical work, so what does that mean for a bacterium? Okay, for us, it's like a muscle contraction. Mechanical work for bacterium, flagella rotor, except it's not ATP hydrolysis, right? It's proton gradient makes the flagellum grow round. What's the other form of bacterial locomotion that we saw? Yes. So these are, that's more like cytoskeleton and muscle contraction, but bacteria, yeah, I'll take that because if you said to me MREB, polymerization, I would agree. That's true. But yeah, I'm thinking about the gliding motion. So this is dependent on type 4 pili, which can extrude and be retracted. And adding subunits onto the base to extrude these pili takes ATP. Okay, so it's required for pushing these structures out from the cell. Mechanical work. Okay, so chemical work, there's something about this that is a little bit of a detail. You could say ATP does all this stuff, but if you look carefully, that's not always true, okay? So anybody know what is used in protein synthesis? GTP is required, yeah. What is used in polysaccharide synthesis? We saw it with the peptidoglycan, if anybody remembers. UDP, yeah, you have starts with UTP, and it's coupled with a sugar monomer to make UDP glucose, or UDP and N-acetylglucosamine, something like that, okay? So UTP for sugars, polysaccharides, and for lipid synthesis, CTP is used. So I've always been quite intrigued by the observation that synthesis of these different essential polymers for cells use different nucleotide triphosphates. So there seems to be a kind of, and this is universal as well. It's not like bacteria do one thing and other types of cells do something else. So this is a universal observation. So probably very early in the origin of life, there was a specialization between which nucleotide triphosphate was used in which biosynthetic pathway. Okay, and so ATP is used for a whole bunch of different things. Okay, and of course they can be interconverted. If you've got ATP and UDP, you can transfer the phosphate group from one to the other, and so you generate ATP and then pass the phosphate groups around. So ATP, important source of metabolic energy for all bacteria, essential for different types of functions in the cell. Redox couples. Okay, no, everybody knows oxidation is loss of electrons, reduction is gain of electrons. And whenever you have a redox reaction, they can be expressed as two half reactions. And now I've always had difficulty understanding why on earth chemists like to do this. But I think the utility is that each half reaction is associated with a redox potential. And knowing the redox potentials of the half reactions will tell you which way the reaction is going to go. Okay? Which out of these partners, which one is going to be the electron donor and which one is going to be the electron acceptor. Okay, so the more negative your redox potential is, the better an electron donor this part of this half of the reaction is. So molecular hydrogen is a very good electron donor because the redox potential is highly negative. NADH is also a good electron donor because this redox potential is negative. So NADH will very easily give up 2 electrons. Okay, so kind of way to think about this is because this is negative, then the reaction will tend to go in this way, in this direction. So when you put the 2 half reactions together, electrons will transfer from donors with a negative redox potential to acceptors with a positive redox potential. And this will give you the delta E, which is the word, what's the word I'm looking for? Difference. It's almost like a potential difference in like if you think about physics, electronics terms, potential difference which will be in volts, right? So overall, if you are using NADH as the electron donor and oxygen as the electron acceptor, then you to calculate this delta E, the potential difference, then you've got the redox potential of the acceptor minus the redox potential of the donor. So for here, it's going to be 0.815 minus 0.3 minus negative 0.32. So plus 0.32. That's where we get 1.14. And it will not surprise you to learn that this redox potential can be, or potential difference, can be translated into Gibbs free energy by this equation, which you do not have to learn, okay? So F is the Faraday constant, and N is the number of electrons that are transferred. And you have a minus sign here, so if I've got a delta E, which is positive, that means the delta G is going to be negative. Okay? So that's why redox reactions will go in one direction, but not the other direction. Okay? Now, I said that NADH or NADPH is a good electron donor. It can give up 2 electrons very easily. Now, why is that the case? So I've put up here the structure of NADH. Okay, so you've got a kind of weird adenosine dinucleotide here. So it's a kind of molecular fossil from the RNA world. And the business end of the molecule is up here with the nicotinamide group. So this is the bit that changes structure in between NAD plus and NADH. So this is NAD plus, and this is NADH. NADH is a good electron donor, so that means the equilibrium state will tend towards this one, NAD plus. So why is this molecular conformation very stable and this one relatively unstable? Yeah. Yep. Yep. Yeah. So what does that mean for this positive charge? Where is this positive charge going to be in reality? Yeah, yeah, it's not concentrated on the nitrogen atom here. It's delocalized around the whole aromatic ring. Okay, so that's why this structure is very stable, because it's very easy to distribute the charge around the ring here. So there's no big, it's no big deal for this group to have a positive charge hanging around. Yeah, so you produce this cyclic structure, which is very good, which is very stable. Good. So that's why NADH, NADPH are good electron donors. Why do you need it? Because a lot of the biosynthetic reactions to make carbohydrate, to make fatty acids, are redox reactions, and they require electrons to be transferred, for example, here onto carbon dioxide. So all of the autotrophic carbon-fixing bacteria need to get NADH so that they can produce carbohydrate from carbon dioxide. Once you've got carbohydrate, you can produce fatty acids. Again, this is a reduced carbon form, right? Think about fatty acid structures. And we saw that nitrate could be assimilated by bacteria after it's reduced to ammonium. And then this is just transferred onto amino acids. So this is very often required. This redox reaction is very often required. Okay, so the last form of molecular or metabolic energy that is present in bacteria and all cells is the electric potential across a membrane. So when you have an impermeable membrane which separates solutions which have got a higher concentration of one ion on one side of the membrane compared to a low concentration on the other side of the membrane, this is, what can I say, kind of form of energy, because if, for example, here the protons which are concentrated on this side of the membrane move across to the other side, this will be an exergonic process. So in general terms, this can be any ion, okay? It can be sodium, it can be potassium, but often, it's protons that there's a proton gradient across the bacterial plasma membrane, and it's an important source of energy for bacteria. So this is the proton motive force. Now, before thinking about the Nernst equation, what is the term? Why is this energetically favorable? If you think about delta G, which of the two parts of Gibbs free energy is being changed here? Delta H or T delta S? Enthalpy or entropy? Entropy, right? Because this is a highly ordered state, okay? If you've got all the protons on this side and 0 protons on this side, there's a lot of order in the system. Whereas if you allow them to diffuse freely, they'll distribute themselves randomly across the membrane. So by transporting the membranes across the membrane, transporting the protons across the membrane, down the concentration gradient, you increase the entropy of the system, okay? So this is entropy driven. And as we saw, it's useful for generating flagellar motion and for transport, for the symport antiport systems. Now, physical chemists have been able to relate concentration gradients of ions across membranes to an electronic potential. So this is the Nernst equation. The only thing that you need to remember about this is that it's going to be related to the ratio between the concentration on one side of the membrane compared to the concentration on the other side. That's going to be important for us later on because it means that if you have a really big concentration gradient, you can generate a really high electronic potential here in volts. And you can use that to drive redox reactions that otherwise would not happen. Okay, I already said this, so yeah, okay. Trophic transport for the jet motion, ATP synthesis by the proton ATPase in the membrane of mitochondria and in the plasma membrane of bacteria that can perform respiration. So in ATP synthesis, the way the energy flows is you start with a redox reaction. So glucose is being oxidized to carbon dioxide. All the electrons are shifted onto NADH. They're then passed down the electron transport chain, eventually onto oxygen. And then this will, to produce water. Then this will, during this process, a proton gradient is formed across the membrane, which will then drive ATP synthesis. Peter Mitchell, Nobel Prize in, I think, 1976, work done in the 60s. Okay, so what you have to remember from this part is, okay, there are the three types of energy currency: ATP, NADH, proton gradients. Whichever they are, you can think of them as kind of money, all right? Dollars, pounds, the euro. It's NADH, but yeah, it's okay. Dollars, renminbi, and and the euro. The three important world currencies. And spending these currencies is exogonic, and that allows you to, do stuff. And they can be interchanged, both theoretically, because all of these things, so ATP hydrolysis, there's a delta G value associated with that, gives free energy. Redox reactions, you've got the electronic potential for electron transfer, which can be translated into a delta G. Proton gradients can be translated into an electronic potential, which can also be translated into a delta G. So in physical chemistry terms, you can interchange these. And also in real biochemical processes, change these forms of energy from one to the other. So if you have ATP, you can use that to generate a proton gradient. And then you can use that proton gradient by reverse electron transport to generate NADH. If you have NADH, you can use that to generate a proton gradient, and then you can use that to generate ATP, if you have a good electron donor that allows you to do this. So if you have any one of these three things, you can get the others. Okay? And that's what allows bacteria to have all these different weird types of energy metabolism. Photosynthetic bacteria, well, they might just generate a proton gradient, okay? Some of them. And then from that, you have to generate ATP and NADH by reverse electron transport. So for all the different types of metabolism we're going to see this afternoon, think about how these three types of energy are getting generated. Okay? And if you can do that, you'll have understood pretty much how all these different trophic types of bacteria function. Okay, so now we're going to start off by looking at glycolysis and fermentation to begin with. So what does this mean? Then it means that you're going to generate NADH and ATP from organic carbon without oxygen. Glycolysis is you start with glucose, 6 carbons, you end up with pyruvate, 3 carbons. And also later this term, you're going to learn the Embden-Meyerhof pathway by heart in biochemistry. But in bacteria, there are also 2 other pathways. Okay, pentose phosphate, we have that as well, in fact. But there are the 3 pathways that can be present in bacteria, 1 or 2 of these pathways. So let's start with Embden-Meyerhof as an overview, okay? We're not going to go through all the intermediate steps and all the structures. In fact, there's going to be no structures here, apart from you just have to remember how many carbons. Okay? So first steps in the Embden-Meyerhof pathway are going to be phosphorylation of glucose to give you fructose 1,6-biphosphate. So the first step is glucose 6-phosphate, right? It's what you get after group transport of glucose into the bacterium. And in order for this to happen, you have to put in 2 molecules of ATP. That's bad, right? The whole point of this is to get ATP. So there's a cost up front. So the cell's got to invest something. Then this fructose 1,6-biphosphate is going to be split into 2 molecules of glyceraldehyde 3-phosphate. This is 6 carbon, 2 phosphate groups, 3 carbon, 1 phosphate group. Now, the next step is, I don't know, probably one of the most important enzymes on the planet, together with rubisco and nitrogenase. And it is glyceraldehyde phosphate dehydrogenase. And what happens here is that, it's a bit strange. Glyceraldehyde 3-phosphate is going to be phosphorylated with organic phosphate, and simultaneously the cofactor for this reaction is NAD+, which will generate NADH, the reduced form. And once you've got 1,3-bisphosphoglycerate, 3 carbons, 2 phosphate groups, each of these phosphate groups will be peeled off and transferred onto ATP. So that gives you a net yield of 2 NADH and 2 ATP per glucose molecule, with pyruvate as the waste product. So that's great, you might think. The cell has performed glycolysis, it's got NADH and it's got ATP. That's what we wanted, to feed the energy metabolism of the cell. But there's a catch. No, sorry, before I say this. All right, so, okay, that's good. So you get these 2 types of metabolic energy. And it all works because of glyceraldehyde phosphate dehydrogenase. Because there's a favorable redox reaction that is coupled to phosphorylation. And that's what allows you to win in terms of ATP synthesis. Now, the other pathways, Entner-Duderov and pentose phosphate pathway, they have different pathways to get to glyceraldehyde 3-phosphate. So the key enzyme in all of these is still GAPDH. Okay, this is the enzyme that's working here, here, and here. What is slightly different here? Here you only put in 1 ATP, so you get a molecule that's got 6 carbons and 1 phosphate group. And when it's split in 2, it gives you glyceraldehyde 3-phosphate and kicks out pyruvate. So you'll still get 2 pyruvate left over. Pentose phosphate pathway, this time you remove a carbon dioxide straight away, so you start off with a 5-carbon molecule and 1 phosphate. And then this is going to give you this aldehyde 3-phosphate, just 1 copy, and then an acetyl group over here. Now, plenty of bacteria will have both the Embden-Meyerhof and the pentose phosphate pathway, or Entner-Dudroff and the pentose phosphate pathway, because ribulose 5-phosphate here is an important biochemical intermediate to synthesize what? No. Close. No. 5-carbon sugar, very important. Yes, ribose. Okay, so you need to get ribose to make RNA, to make DNA. And ribulose 5-phosphate is the kind of feed chemical to in this pathway. So very often the pentose phosphate pathway exists, but it's not really used as energy, as a pathway of producing energy in our cells, for example. But there are some bacteria which just have the pentose phosphate pathway. They don't have Entner-Dudroff, they don't have the Embden-Meyerhof pathway. Okay, so this one will be used for generating metabolic energy and also generating the intermediates for synthesizing ribose. Okay, so the basic energy characteristics of all of these glycolytic pathways is that you get a couple of molecules of NADH or NADPH, and then 1 or 2 molecules of ATP per glucose that you put in. And that's a problem, because in the energy economy of the cell, you need a lot more ATP than you need NADH. So this is not enough. The yield is too low. So you might think, well, that's no problem. Look, all we have to do is just keep on putting more glucose into the system. Even if we get ATP, and if we only get 2 ATP, it doesn't matter. That just, lyse more glucose. Keep putting it in there, and you'll get enough ATP. But that can't work, because NAD+ is an essential coenzyme for the step that allows you to generate ATP. So if all of your NAD coenzyme has been reduced into NADH, you can't put any more glucose at the top, because this step will not work. You need to regenerate NAD+ in order for GAPDH to function. So that's the problem with these just having glycolysis. Glucose as the input, GAPDH to generate ATP. You need to get rid of the excess electrons that are all stuck on NADH, because you need to get rid of them to regenerate the oxidized form, NAD+, so that you can carry on lysing glucose. So NADH is the electron donor, and you need to find an electron acceptor. What can you use? Well, you've got a lot of pyruvate. This is the waste product of the glycolytic pathway. So fermentation reactions are basically a solution to regenerate NAD+ by putting the electrons onto something that's derived from pyruvate, because you have it in the cell. It's lying around. You're going to throw it out anyway. So use it for something before. So that's really the point of fermentation. Glycolysis, you'll generate pyruvate and NADH. And somehow what you want to do is transfer the electrons here onto the pyruvate. So if you do that directly, that will generate lactic acid. That allows you to recycle and get NAD+, which you can then use in this step here. And that allows you to generate ATP. That's it. The problem is solved. And in that case, yeah, you can keep on putting glucose into the system. Fermentation pathway will just run around with the recycling NADH2 NAD+, and you generate 2 ATP every glucose molecule that gets degraded. Easy. So that's it. Now what we're going to do now is look at the different types of fermentation pathways that bacteria can use, because they don't all do the same thing. So this is a kind of a simplified scheme of a diagram of different fermentation pathways in bacteria, and we're going to look at them one after the other. And what you have to remember is that at some point, pyruvate is always the electron acceptor. Okay. Oh, and for all of these, you should remember one example of a bacterial species that performs this type of fermentation. Okay. Right, so we'll start off with the simplest ones. So pyruvate, that's this here, can be reduced to lactate. So you have this keto group here, maldehyde group, reduced to alcohol group. Lactate dehydrogenase. Very simple reaction. One step, lactobacillus, streptococcus, lactococcus, used for making cheese and yogurt. Okay. Lactococcus lactis, streptococcus thermophilus, lactobacillus bulgaricus. These are species used in making food. Lactic acid fermentations can be called homo or heterolactic. Homolactic, lactic acid is the only fermentation product. Heterolactic, it's lactic acid plus something else. Okay. So very important in the food industry. Ethanolic fermentation, two-step reaction. So first, pyruvate decarboxylase is going to kick off carbon dioxide from here, right? This is just going to leave as a, no, this one. This is going to leave as a carbon dioxide. And then this acetaldehyde will be reduced to ethanol. Mostly, this is Saccharomyces cerevisiae, yeast is performing this reaction. For brewing and baking, you get carbon dioxide and ethanol as the fermentation products. Zymomonas is a bacterium that performs the same reaction. Okay, so these fermentation pathways are very simple. They allow the bacterium to carry on generating ATP by fermenting more and more glucose. But they both have a problem. What happens if you accumulate too much ethanol in your body? What happens if you put a bacterium into 70% ethanol? It will die. Okay, it's toxic. Ethanol is a toxic waste product. What happens if you have a load of lactic acid building up? pH is going to go down. And low pH is going to be harmful for bacterial growth. So the drawback of these simple fermentation pathways is that they generate toxic waste products, either because they're acidic or because it's ethanol. So there's a limit to how much lactic acid that a bacterium can pump out. That's why these are very useful for conservation, right? Because when you make yogurt, only the lactic acid bacilli can can survive, and no other bacteria that are there will be able to grow in the yogurt. So it's a biological food conservation technology. Okay, so often the more complex pathways are ways to limit the production of toxic waste products. Okay, so let's go and look at slightly less simple pathways. So you start again with pyruvate and stick together two molecules of pyruvate, kick out the carbon dioxide. So you start with 2 times 3, 6 carbons, lose one here, alpha acetolactate, this is 5 carbons. Kick out another CO2, acetoin, 4 carbon molecule, which will be reduced to butanediol. Okay, don't need to draw out these structures. Everybody can imagine 2,3-butanediol, right? Well, I'm just asking. All right. So this is quite common. So it's like Serratia Enterobacter bacillus. So these are going to be type of bacteria that live in people's guts, gram-negatives here, gram-positive there. And it used to be economically very important because butanediol was the starting chemical for the synthesis of artificial rubber. So the fermentation pathway used to be exploited industrially, but now it's petrochemicals that are used. So if ever we need to, then we could revive this fermentation pathway to produce a biological artificial rubber, if that makes any sense. Now, to understand the other fermentation pathways, you have to have an understanding of where pyruvate can also be used in other types of metabolism in the cell. Okay, so pyruvate can be carboxylated to give you oxaloacetate. So 3 carbons here, add 1 carbon, you get 4 carbons compound here. And this is important for many pathways of amino acid biosynthesis. So many bacteria will have this kind of pathway. Facultative anaerobes. These are bacteria that can perform respiration in the presence of oxygen, but will be able to survive by fermentation without oxygen, in the absence of oxygen. Now, in that case, these bacteria will perform the tricarboxylic acid cycle, the Krebs cycle, as part of respiration. So they will also have a pathway that allows them to generate acetyl coenzyme A, and this will feed into the tricarboxylic acid cycle. So keep that in mind for 1 second. And this will help us to understand why on earth Propionibacterium perform this fermentation pathway. You start out with pyruvate and you kick out propionic acid. It's 1, 2, 3, 4, 5 enzymatic steps here. You say, why are they doing something so complicated? Why don't they just use lactic acid fermentation and just produce one enzyme? Well, the answer is because, okay, this one, pyruvate carboxylase, they already have it. And these enzymes here, oxaloacetate to malate to fumarate to succinate, these are Krebs cycle enzymes that are running in reverse. So for other types of metabolism, these bacteria already have these enzymes. And this will give you propionic acid as the fermentation, as the waste product, and not lactic acid. Can anybody else see an advantage in doing this compared to lactic acid fermentation? Yeah.
You already have all these enzymes, so you don't need to produce another one.
Yeah, we need to produce this one maybe. We need to produce one. But it's the same thing with lactic acid fermentation. LDH you need to have. Anyone else? Here you are using 2 molecules of NADH. Okay, so you use up 4 electrons. So you recycle twice as much NAD for 1 molecule of pyruvate, and you only get 1 molecule that's acidic. So in terms of how much the pH is going to go down, it's going to be less drastic. So the problem of ejecting a waste product that brings the pH down is mitigated a little bit. So propionic acid fermentation, propionibacterium. That one's easy to remember. Now E. coli and a whole bunch of other gram-negative enterobacteria will perform mixed acid fermentations, which is the kind of horribly complicated one. So to try and make this simpler a bit for myself and hopefully for you too. Now, remember, earlier on I just presented the transformation of pyruvate into acetyl coenzyme A, and this is going to be useful for fatty acid synthesis, for example, or the Krebs cycle. Problem with using this step here, if you use pyruvate dehydrogenase, that's not going to work very well because you're generating another molecule of NADH. And the whole point of fermentation is to get rid of that and regenerate NAD+. So in a mixed fermentation reaction, mixed acid fermentation, you don't want to use this pathway. So in E. coli, for example, you start with pyruvate, and it's another enzyme which is called, let me just read my crib sheet here. Pyruvate formate lyase. Okay, that's right. So you get pyruvate is going to be cleaved into formic acid and acetyl coenzyme A. So this is one waste product, formic acid. Acetyl coenzyme A is then going to be transformed into something else, a mixture of all these different fermentation products which are going to be produced as well as formic acid. Now, all of this is very complicated. Okay, so that's where the name comes from, mixed acid. Okay, so you've got formic acid plus a mixture of other fermentation products. Can be ethanol, can be isopropanol, butanol. So this is important because it explains why everybody walking around has got a very, very low level of ethanol circulating in our bloodstream. We do not have 0.000 grams per liter of ethanol because E. coli in our guts are performing this mixed acid fermentation, and there's some ethanol produced. Okay, E. coli and other bacteria in our guts. And there are some people who have a rather unusual gut flora, and their bacteria produce a lot of ethanol. So if they eat a kind of a meal that's rich in sugar or starch, then they can actually get drunk. And there's been legal cases where someone's been pulled over for like driving like this in their car. And the police have given them an alcohol test, a breathalyzer. That's it, they're over the limit. They're driving while under the influence of alcohol. And the person who was arrested, yeah, but I don't understand, I didn't have anything to drink. And it was actually true. Of course, the police officer was, yeah, I don't hear that one every day. But it was, for some people, it's actually true. And they have this very, very particular gut flora, which is producing ethanol in their guts and can make them drunk. So they have to be careful what they eat. Okay, so apart from the anecdote here, so ethanol can be produced, and some of the other branches of the mixed acid fermentation pathway are interesting because you can scrape out another ATP molecule. Okay, so if you're thinking about, well, why does E. coli use such a complicated pathway? The answer is it allows the bacterium to, get a couple more ATP molecules. So it's worth it. It's worth doing something complicated because they gain in terms of, generating ATP. Now, once again, the big problem for E. coli and other bacteria using these pathways is the production of formic acid. So the pH is going to go down, and this will inhibit bacterial growth. And when that happens, then they will cleave formic acid into molecular hydrogen and CO2. Now, which of these products will be released depends on the pH of the medium. So if you grow E. coli in the absence of oxygen with glucose as a substrate, it will perform mixed acid fermentation. And these are the different fermentation products. So you get a lot of formic acid, quite a lot of ethanol, quite a lot of lactic acid, acetic acid, and a bit less of all these guys. When the pH goes down, E. coli will start to cleave the formic acid into hydrogen and CO2. So these products go up. Then in your guts, or the guts of animals, hydrogen and CO2 can be used by methanogens to produce methane. So that's where the methane comes from in, well, our animals' lower intestines. Okay, so think about this. There's more than one correct answer. Okay, everybody got it? Okay, energy yield per carbon is low. This is a problem. Hands up who says yes. It requires oxygen. This is a problem. Who says yes? Only used by gram-positives. Anybody say yes? The end products can be toxic. Okay, everybody's remembered this one. Requires cytochromes. Nobody understands what that means. Okay, no. Okay, yeah, that's true. End products can be toxic. That's correct. Only used by gram-positives, no. Gram-positives and gram-negatives can perform fermentation. Yeah, but this one, everybody forgot this one practically. Okay, more than one correct answer, I said. So this is a problem. It's a limitation. Energy yield per carbon, per glucose molecule is low. So you need to put in a lot of glucose for bacteria to grow by fermentation. Glucose or another organic carbon source. Okay, that's where we got to by now. So I guess it's a little bit early, but I think this would be a good time to take a break, because then I'm going to go on to respiration and other forms of energy metabolism. So we'll take a break now and then come back for a superpowered 2-hour session afterwards. Okay, hour and a half, a bit more maybe. Yeah, well, it will be, because we're supposed to have 2 hours, 40 minutes this afternoon. We've had 1 hour, so there's another hour and 40 minutes, potentially. Okay, so everybody go take a break.